Welcome back to my channel Knowledge Chronicle.
Have you ever wondered what makes you, you? What is that magical code inside every living cell that decides your height, your hair color, your eye shape, and even your talents? That code is DNA, deoxyribonucleic acid. And today we're going to unlock the entire mystery behind it, from its discovery to its structure, to how nearly 2 meters of DNA fits inside a microscopic cell nucleus. Get ready for a deep dive into one of biology's most powerful molecules. Let's begin. What is DNA? DNA stands for deoxyribonucleic acid, a long-chain polymer made of repeating units called deoxyribonucleotides. Now, what does that mean? A nucleotide has 3 basic parts. 1, a nitrogenous base. 2, a pentose sugar deoxyribose in DNA. And 3, a phosphate group. These nucleotides are joined end-to-end to make a long strand, a polynucleotide chain. But how long is DNA really? Let's take some examples. A bacteriophage, Phi*174, has about 5,386 nucleotides. Bacteriophage lambda contains 48,502 base pairs. The E. coli genome has 4.6 million base pairs. And a single haploid human cell has around 3.3 billion base pairs. That's right, 3.3 billion. This length is a fingerprint of the organism itself. DNA versus RNA. Here's a quick comparison. DNA uses deoxyribose sugar and has thymine. RNA uses ribose sugar and has uracil instead. Also, every nucleotide in RNA has an extra OH group at the 2' carbon, making RNA less stable but more versatile. The discovery of DNA and double helix. DNA was first discovered in 1869 by Friedrich Miescher, who named it nuclein. But it wasn't until 1953 that the most revolutionary discovery happened. James Watson and Francis Crick, using X-ray diffraction data from Rosalind Franklin and Maurice Wilkins, proposed the double helix model of DNA. This model was strongly supported by Chargaff's rule, which stated amount of adenine equals thymine, amount of guanine equals cytosine, which means the strands are complementary. If you know one strand, you can predict the other. This also explained how DNA replicates. Each strand acts as a template to form a new one. So from one DNA, you get 2 identical copies. This is how genetic information is passed on. Features of the double helix. Let's look at the key characteristics of the double helix. First, there are 2 strands with sugar phosphate on the outside and bases on the inside. The strands run anti-parallel, one from 5' to 3' and the other from 3' to 5'. Base pairing is next. Adenine pairs with thymine through 2 hydrogen bonds, and guanine pairs with cytosine through 3 hydrogen bonds. The helix has a right-handed helical structure. The pitch of the helix is 3.4 nanometers with about 10 base pairs per turn, and the distance between base pairs is 0.34 nanometers. Base stacking helps stabilize the structure. Because of the purine pyrimidine pairing, the width of the helix stays uniform. Imagine a spiral staircase with perfectly equal steps. Central dogma. The double helix led to the famous central dogma of molecular biology. Proposed by Francis Crick, DNA makes RNA and RNA makes protein. But in some viruses like HIV, the flow goes backward. RNA makes DNA. This process is called reverse transcription, DNA packaging. Here comes the big question. How does 2.2 meters of DNA fit inside a nucleus that's only 10 micrometers wide? In prokaryotes like E. coli, DNA isn't in a nucleus. It's located in a region called the nucleoid. It's held together by proteins in loops. In eukaryotes like humans, DNA wraps around histone proteins to form nucleosomes. Each nucleosome is DNA plus a histone octamer, which consists of 8 histone molecules. About 200 base pairs of DNA wrap around each nucleosome. Under a microscope, these nucleosomes look like beads on a string. This is the chromatin. Chromatin further coils into chromatin fibers, and during cell division, these condense to form chromosomes. There are 2 types of chromatin. Euchromatin, which is loosely packed and active in transcription, and heterochromatin, which is densely packed and inactive. And for even higher packaging, non-histone proteins, or NHCs, help fold DNA into supercoiled loops and chromosomal structures. Let's calculate the DNA length of E. coli. It has 4.6 million base pairs. So, 4.6 times 10 to the 6th base pairs times 0.34 times 10 to the negative 9th meters equals approximately 1.56 millimeters of DNA, packed into a cell just a few micrometers long. Also, a human cell with 3.3 billion base pairs has approximately 5.3 times 10 to the 9th base pairs times 0.34 times 10 to the negative 9th meters equals approximately 2.2 meters of DNA in every single cell. And guess what? Your body has approximately 37 trillion cells. Imagine the total DNA length in your entire body. It could stretch to the moon and back multiple times. So DNA is not just a molecule. It's a library of life. It holds instructions to build and run you, cell by cell, organ by organ. From its discovery to its elegant double helix and to the incredible packaging within a tiny nucleus, DNA continues to amaze scientists every single day. If you found this video helpful and engaging, don't forget, your one like, share, and subscribe will help us create more valuable content. This is Knowledge Chronicle, and we'll be back with more fascinating science that makes sense.